Okay guys, just to wrap up this module 3, let's talk about what some of the mechanisms are by which bacteria are able to protect themselves from antibiotics that exist out there. All of these different classifications we talked about. You may recall this statement that I shared with you last time. There was a thought in the mid to late 1900s that we were going to just get rid of some of these bacteria. They're going to be a thing of the past. And now we know that's absolutely not the case. So I just wanted to share with you a little bit of the history moving on from 1940 when we had some of our penicillins that we were producing. We're looking at World War II, ramping up production of that, of Fleming's penicillin. And over here you see on the right antibiotics that are being introduced over the years and the resistance that antibiotics are developing over the years, or at least resistance that we have been able to identify, right? Because I'm sure there's resistance out there. These cells have been warring against one another for millennia, right? Using these antibiotics because they produce them. So here you can see how it is that we have identified lots of antibiotics over the years, but then we would identify also some of these organisms that would crop up that had resistance. Now, as we go through the years here, and now we're going on past 1965, we're going into about when I was born, around here, right, in 1969. I know that sounds ancient to some of you, but we're still identifying drugs, okay, over on the right, but then we're seeing more and more and more of these resistant forms of bacteria cropping up, all right? Let's move on into the 2000s. Now, I know some of the people in my class are born around then, right? Made me feel really old when my classes started to have birth dates of 2000 in there. But here we have now a few drugs we're identifying, but a whole lot of drug-resistant forms. And you notice that these are multiple, like XDR tuberculosis, that's multiple antibiotics that tuberculosis is resistant against. And this is happening more and more. We see lots of bacteria that don't just have one resistance, but they have multiple resistances, all right? So this is a major problem. It's not like these antibiotics are winning out over the bacteria. And we have to keep in mind that bacteria are always going to evolve faster than we are, and faster, most likely, than we can produce drugs against them, all right? We need to be smart about it and understand it, and keep in mind that these are the pathogens we're focusing on. When we apply antibiotics, we're also affecting the normal flora that exist on us and in us. So we have to be mindful of these things, all right? The prevalence of resistance among bacterial strains, I made this graph from some data I found, and this was over a 25-year period. It's a little dated now because this was like 1975 to 2000, but it's really telling. If you look at what were the percent of bacteria of these different species, like E. coli here, what was its level of resistance to this sulfa drug, trimethoprim? Well, in 1975, 0 were resistant. That drug would kill every E. coli around, all right? In 25 years, it went up to 28%, okay? Staph aureus, tetracycline would kill everything but 3%. There were 3% of Staph aureus that were resistant. Now, well, in 2000, I should say, almost 30%, almost a third of them were resistant. Tuberculosis, only 1% would be okay around the antibiotic that you see there. Now 35% are resistant. Pneumonia, so this is strep pneumonia. When it came to penicillin, only 1% were resistant. Pretty good drug to use against pneumonia. now 40%. This is a really interesting one. Neisseria gonorrhea, right, causes STIs. Tetracycline would do a great job on gonorrhea, and it would kill all but 2% that may be resistant. Now 75% of Neisseria gonorrhea are resistant to that drug. Not a very good choice. You better consult those tabulations, right, that they keep, databases that they keep, and choose a different antibiotic, all right? And these are a little more recent updated information on the gravity of the issue and the amount of deaths that are attributable to antimicrobial resistance. And this is a forecast of what they think there's going to happen by 2050. Here we have, just for comparison's sake, what tetanus, what the deaths are that are caused by tetanus, okay? And this, here we see cholera, we see measles, how many people are typically killed by these particular diseases. Road traffic accidents, right? Pretty big. Antimicrobial resistance is pretty low right now, but they think, based on all the information that's coming in with antibiotic resistance and our relative difficulty with producing new drugs to treat these antibiotic resistant bacteria, they think that this is going to be the size of the circle by 2050. We're talking more people dying from antibiotic microbial resistance than cancer. Pretty eye-opening, right? All right, so when we talk about antimicrobial resistance, which is a decreased susceptibility to an antibiotic, right? So if I apply an antibiotic and it kills everything, they're very susceptible, right? If I apply it and 75% of them still survive, then there is a decreased susceptibility, right? They are resistant, in other words. So let's introduce a couple new terms. Cross-resistance. It is possible for a single mechanism to confer resistance to several different antimicrobial agents. Put differently, a single mechanism, so let's say a bacterium has a gene, and that gene results in a protein that performs in a way that will give the bacterium resistance to similar drugs. So let's take, for example, the beta-lactam subgroup of cell wall antibiotics, right? Penicillin, ampicillin, oxacillin, you take the whole group. Imagine a single gene that produces an enzyme that cuts down every one of those cillins, right? Breaks down penicillin, ampicillin, any of those beta-lactam drugs. That would be a cross-resistance. So a single mechanism or gene that confers resistance for that bacterium to lots of similar drugs, okay? Cross-resistance across that entire similar category, there's protection. Now, if we talk about multiple resistance, here we have multiple mechanisms that confer resistance to several different types of antimicrobial agents. So let's paraphrase that. Multiple mechanisms, to me, I think genes. So multiple genes producing multiple different enzymes, which give you the ability to break down different kinds of drugs, very different kinds. So let's say you had a gene that makes an enzyme that breaks down all beta-lactams, like we just talked about. But then also, that bacterium also has another gene that confers resistance to a whole subgroup of protein synthesis drugs. Multiple resistance. An example of one that we already talked about here is R100. R100 was a plasmid. It's found in E. coli and a bunch of other bacteria, and it has several different genes that confer resistance to different kinds of antibiotics. That would give a multiple resistance ability to any cell that got that plasmid. Okay? Now, how is a bacterium going to acquire such a mechanism, such a gene, right? How does it appear in the first place? It's possible for mutation to change things, right? Mistakes that are made, or certain environmental pressures that alter the DNA. And we've talked about point mutations and things like that that could really change things. And this mutation of an existing gene could give resistance, right? It could change how susceptible a bacterium is, right? So suddenly this organism has a gene because of mutation that would give some protection. So this is one way it could happen. But let's think about this. Mutation like that, maybe radiation causes a mutation. That kind of mutation is rather random. And if I had to, an analogy I would use for this is if you just spin me around in circles with a blindfold on and I throw a dart, okay? Am I going to hit the board even? Much less the bullseye. If I hit the bullseye, that would be a good result, right? So a good result for the bacteria here would be acquiring the gene, having a mutation that gives you the gene for protection, right? For this antibiotic resistance. The likelihood is slim if we have random mutations that cause changes like that. However, the acquisition of new DNA on plasmids, on transposons, these horizontal transfers we talked about, that's far more likely to give the cell what it wants, what it needs, okay? So one of these resistance genes being passed is far more likely. And we know about some of those methods, right? We've got transformation, conjugation, transduction. All of these could pass an already existing gene for protection to a living cell. Now this image here actually I think is a really good one from another text that actually it's online, I found this one, and I think this is just really good because it really dives into everything I would want you to know about how these mechanisms protect. Okay, so let's go through each of these. There's 6 of them. Okay, I'm just going to talk through each of them with you. The first 2 of the 6 mechanisms by which this bacterium might protect itself from an antibiotic, I would say have to do with the quantity of the antibiotic that accesses the cell and has its effect. Okay, so here we have an antibiotic over here, this little circle here. Okay, and the first mechanism that we could talk about is some kind of characteristic about the cell membrane or the cell wall that reduces the amount of antibiotic that can enter the cell. So a gene codes for a particular ability to keep the antibiotic out. Reduced uptake, number 1. Okay, so first aspect that could protect. Now related to that, but because it has something to do with quantity that can access the cell, let's say it does not reduce the uptake, the antibiotic gets in no problem. Well, if this cell has a gene that codes for a pump in the envelope, in the membrane, let's say, of this cell, and it's constantly pumping the antibiotic out actively, well, this will also reduce the quantity that has access to the inside of the cell, right? It gets in, but it's pumped immediately out again. So this would be an efflux pump, okay? And so we have number 2 here, and you see how they're related, right? Because they both have to do with how much antibiotic is in there. to kill the cell. All right, the next two mechanisms are also kind of related. So here we have the antibiotic down the bottom, and here they show it getting into the cell no problem. Okay, and they show that up here as well. It gets into the cell, no problem. All right, so what might happen to that antibiotic once it enters the cell? One thing that could happen, and you can see it here with the green arrow, there could be a gene that codes for an enzyme that alters the shape of that antibiotic. So this circle becomes a square here, right? As an altered shape, this molecule will not likely behave and cause the death of that bacterium, okay? If it's a different shape, it's going to have a different function and probably isn't going to function the way it used to. So we have an antibiotic altering enzyme that is coded for by the cell. So third mechanism, okay? Okay, somewhat related to this might be an antibiotic degrading enzyme coded for by a gene. So it doesn't just change the shape, it totally obliterates that antibiotic. Not going to kill the cell now, right? Because it just destroys it. So an antibiotic degrading enzyme, fourth mechanism. All right, last set of possible ways we could interfere with the antibiotic function, okay? And these have to do with the metabolism of the cell. So let's say this antibiotic up here in the top right now is a sulfa drug. Let's say it interferes with folic acid synthesis. This is critical for a bacterium to be able to replicate and grow, okay? Remember, folic acid is used by bacteria to make, nucleotides, right? So this cell can't even replicate its DNA if this antibiotic is going to do what we want it to do. So here we have, let's say, a process that's critical to the bacterium. X becomes Y. There's an enzyme XY that mediates this reaction. So let's say Y is folic acid, right? There's some substrate that needs to be changed into folic acid, and this antibiotic interferes with XY. It interferes with the production of folic acid, okay? Think of Y as that folic acid that we need to produce. And this is how sulfa drugs kill, right? You can't get your folic acid, you can't grow. What if this cell produces so much XY enzyme, more of the enzyme than the sulfa drug can neutralize? Right? The sulfa drug is a competitive inhibitor. It'll stick its nose into all of the active sites that it can. But let's just make up a number. If there's 1,000 XY enzymes and only 500 sulfa drugs that get in, then there's 500 left to continue to mediate this reaction. The cell is going to still grow. Okay? So, overproduction of the target enzyme. This is a fifth mechanism that the cell may use to get around this metabolic inhibition. All right? Finally, let's go back to that idea, right? We have this XY enzyme critical for folic acid production. We toss in a sulfa drug. The sulfa drug, let's say the XYs are all saturated. All of those enzymes are interfered with. But let's say there's a gene within this bacterium that allows folic acid and nucleotides to be produced in a different manner, using different enzymes like an XW enzyme, brand new kind of enzyme that sulfa drugs don't work on. Okay? That would be a bypass around the original pathway, right? So we're going to have a sixth mechanism, a metabolic bypass. Okay? So 3 really interesting ways that antibiotics protect themselves. These are like, a suit, a protection, right, from all of this artillery that's around, all of these antibiotics. Okay, this is like a suit of armor, all of these protections. Now, what do we need to do? Now, understanding that these resistances can be shared very easily in a powerful way. They can share through conjugation, transformation, right? Even through viruses we've seen, through transduction it can happen, right? So what do we do about it? We need to be smarter about it. The misuse of antibiotics is the problem, is a big part of the problem, okay? It's true that resistances develop and evolve in bacteria naturally because these microbes use those antibiotics on one another without our help, okay? But we just exacerbate the problem, okay? Every time you take an antibiotic, you choose for the strong ones. You kill the weak ones, the susceptible bacteria, but the others that have the resistance, they continue, okay? And this goes for any ecosystem. It could be the ecosystem of your body that you're thinking about, but it could be antibiotics that are in the soil, right? What are you going to do with antibiotics that you are you have extra antibiotics left at home, right? You were instructed not to take certain antibiotics. Let's say they caused some allergies for you. Well, I wouldn't flush those down the toilet, right? I wouldn't want to do that because ultimately that makes its way back to the soil, and the soil has bacteria, right? And you're basically adding to the problem in that respect. So what we really need to do, we need to avoid overprescription and unnecessary prescription of these antibiotics. It's absolutely true that if you have a viral infection, you should go away without an antibiotic. If they know that it's viral, they should not give you an antibiotic. Now, there are situations where you might have a viral infection that develops into a bacterial infection. You start with bronchitis, but then it becomes a pneumonia that's caused by a bacterium, right? Because you can't, maybe that mucus that's in there you can't get out, and then the bacteria gets stuck in there, right? And you have a different monster at that point. But it's not a good practice to just throw drugs at any viral infection. You don't want to use antibiotics without limits with animal feed, right? Because these antibiotics, again, they add to the problem, okay? You don't want to pause taking drugs in a prescription, right? So if you're given antibiotics to treat a bacterial infection, and you should take the entire course, right? If you, let's say out of a 100% infection, right? 100% of cells that are causing an infection, only 10% of them are resistant. Well, the drug you're given may kill the 90% that are susceptible. The other 10% your immune system can handle, all right? But if you start feeling better after 3 or 4 days and you stop taking the drugs, well, you won't kill all of the 90% that were susceptible. You'll leave a good bunch of those behind. And guess what? Those that were resistant can share their plasmids with those that are susceptible, and now they're resistant. You go back, you're feeling bad again in a couple 3 more days, and you find that, oh, I can't go back on that same drug. I now have a resistance that's been shared because of this horizontal transfer that we've gone over, all right? I mentioned this already. You don't want to flush stuff down the toilet, right? You don't want to have improper disposal of antibiotics. And this slide here kind of goes through some of the issues we're having when it comes to antibiotics. All right. Some of them I already mentioned, right? Overprescribing, not taking as prescribed, right? Stopping taking your drugs. Antibiotics in agriculture, we already talked about that. And these are some others I didn't actually touch on, but we need proper tests, right? We need to know, is it viral or bacterial? We need proper hygiene. And of course, we want to be careful in the hospitals and ICUs and places where sick people go, right? We want to be particularly careful and smart about it. So any nurses in the class, this is not to make you paranoid. This is not meant to do that, but it's meant to bring your attention to how cells grow, how they share their resistance, and to be smarter about it, right? We're not going to beat bacteria when it comes to their evolution. They evolve faster than us, but we can be smarter about how we prescribe, how we behave around a patient who is infected with particular types of bacteria to control their spread. All right? All right, guys, I think this is it. This is all we've got for this particular big Module 3. And everything should be up there now for that test, the next one coming up.